So it's my pleasure to introduce Phil Jones. Phil is the Vice President of Therapeutics Discovery at MD Anderson and the Head of Drug Discovery at the Institute for Applied Cancer Science. What is not listed in his bio is the fact that he's also my boss. So, in case you're unfamiliar with the Institute for Applied Cancer Science, we're basically a biotech embedded within MD Anderson. So we're on the south side of the main campus of MD Anderson. And basically, you could say, well, what makes you different from any other biotech you may find if you walk down the street of Cambridge or Boston or the Bay Area. And what makes us different is our direct connection to the clinic, of course, and we try to take full advantage of that as much as possible. As Michelle was just nicely alluding to, it's great very early on a project if you know exactly what the needs are in the clinic and what that patient population may be going forward from the beginning. And we're fortunate to have that type of direct connection. And with that, I'll hand it over to Phil.
Thanks, Mary. So I think Michelle's done a fantastic job opening up the session, and I'm going to reiterate on a lot of points Michelle has made. So I'm going to say them from a slight different perspective, but the fundamental philosophy that Michelle has said is going to be kind of at the core of what comes through the talk in the next 25 minutes. So the reason we're all here today is really we're going to try and do an experiment in humans. We're going to really test the hypothesis that changing the abundance or activity of a particular target, we typically think a protein, but we could also be modulating an RNA, a DNA in some way. You're going to interfere with that in some manner, and that will actually have a direct impact on the disease or ameliorate the symptoms of a disease. And just as we're here in this forum, that can actually mean any form of intervention. Michelle has focused very much on small molecules, but you could come in and you could modulate the disease by a peptide. You could come in and modulate that with some form of protein therapeutic, whether it be an antibody, a Fab, or kind of some bispecific modality. We're seeing the emergence of cellular therapies, and we will see it with time, genetic reagents as well in the clinic. So just to level set the playing field here as well, I'd like to ask you a question like, why are you all here in the room today? What are you hoping to get out of this series of talks? Are you trying to really solve an unmet medical need? Are you trying to solve a medical problem? Are you really trying to find a home for your science? Are you trying to understand whether your science could impact a disease? Are you here to really make money? All of these are really noble goals, and there's a good reason why you could be sitting in the room, but each of these has its own unique challenges associated with it. The first one, this is really, as Michelle said, the simplest of all of it. You know exactly where you're trying to go. The road is there in front of you. You know which of those patients you want to treat when you get to the clinic. You know that you modulate that particular target, you're going to change those patients. The big challenge of that is, how on earth are you going to do that? Are you going to do that with a peptide? Are you going to do that with a small molecule or protein therapeutic? What do you actually need to be building here to go forward? The second one is a lot more tortuous path. I mean, how are you actually going to you want to climb to the top of the mountain here. You want to actually show that your science will have an impact on the disease here. That is really, really difficult. And the best way to achieve that is, as Michelle said, go out and talk to other people. Find yourself 10, 20, 50 different clinicians. Talk to them about your disease, talk to them about your biology, and hopefully there may be a meeting of the minds and you may be able to find it. However, you have to be realistic here. You may not have the tool here. There is no way that you're going to get your NASCAR up that mountain, however much you try. And the third point here, if you're here to make money, well, all I can say is you may want to go somewhere else. This is going to be a lot tougher than what you want to do here. So why can I actually stand before you and talk to you about this? It's really because I've lived this business for the last 20 years or so. So I'm going to try and get 20 years experience into 20 minutes of slides here. I'm a trained chemist. I got my PhD in the UK in Nottingham. I then really had the fortune of working for Merck really across the globe. Worked in 3 continents, really worked on a bunch of different projects. And I really got to learn this drug discovery paradigm. I saw programs from HTS screens here, as Michelle was just saying, where we screened 3 million compounds. And I've been fortunate to see compounds get it right the way across the marketplace. And as Mary said, came here to MD Anderson back in 2011 and built up the IX group. So I really chose to use my scientific background to try and make an impact on people's lives. So I do this by not trying to solve things myself. I go out and talk to a multitude of people, find people that know more about a human disease than me, and then join forces with them. Because together, and this I think is one of the things that you want to get from today's seminar series, is that you're not going to solve this on your own. You can be really, really smart, but you need to find people smarter than you to succeed here. So this is one of the slides that you kind of always used to, whenever Merck used to have a symposium, you always used to get to see this quote presented. Never try to forget that medicine is for the people. The profits will follow. The better we have remembered that, the larger those profits have been. So really, if you can solve our medical needs, profits will follow from that. And luckily, I've kind of worked on 2 projects in my life that have actually had that. At the turn of the century, we started at Merck working on an HIV integrase inhibitor program. At that time, you had HIV protease inhibitors, you had NNRTIs approved. Patients were relapsing to those. People were dying of their AIDS. Merck came along and realized that if you inhibited the viral integrase enzyme, you could have an impact on disease. We developed this molecule, raltegravir. Last year, it sold $1.4 billion of annual sales. The second program I've been and worked on is the PARP inhibitor, niraparib. This really came out from the understanding that there are genetic mutations out there that drive breast and ovarian cancer. And if you inhibit the PARP enzyme in those patients, there will be a disease-modifying impact. And again, that was actually approved back in 2016. So what's it going to take for you? Well, it's really going to take a lot of your time. It's probably going to take you 7, 8, 9, or maybe even more than a decade to actually get your projects all the way to the clinic and show they work. It's going to take money, and it's going to take a lot of sweat and tears to make that happen. But the way you can succeed is if you kind of really remember a couple of underlying tenets here. So number 1 is keep your eyes wide open. Really, as you're doing your science in the lab, pay attention to that data. That will lead you in a certain direction, and really be critical of your own data, and scientific rigor is key. The second one is really stay focused on what you're trying to achieve here, and ask yourself, is your problem really going to be able to solve that unmet medical need that we want? The third people is you need to be prepared to get stuck in and roll up your sleeves, get work done, but also learn. Drug discovery is a learning process. It's about learning about your program, about how your program has an impact on disease, but it's also about learning the process and making sure you don't make mistakes along the way. And to do that, you really need to surround yourself with a bunch of people. The group down at in MD Anderson South Campus is this group on the slide here. We're about 100 people. Again, it takes about 20 or 30 people working on the program at any one time to really succeed here. And you need to be able to tap into people that know about particular disciplines to help you succeed here. And then again, rigor is going to come through time and time again. And I know that the GCC has many seminar series about scientific rigor, and it is important. And I think one of the key tenets to really remember is here, with your program, would you really treat a loved one with your drug? If you kind of hold that near and dear to you the whole time and actually think, would you treat your sister? Would you treat your mum and dad with the drug that you're thinking about developing? That will really give you some level setting on how much you value your own program. And also be prepared to pivot. Projects take surprising turns along the way, and that can also quite often work to your advantage. So the other thing to remember is you're not working in a vacuum. There are many people that are trying to solve the same medical need as you. And so this is actually a plot of kind of the standard of care for non-small cell lung cancer, going back here from the 1940s to where we sit today. And you can see that this was a pretty level playing field, right the way up to probably about the turn of a decade here. And at that moment in time, you had projects like Alotinib, the EGFR inhibitors, that were approved because they gave a change in overall survival here of, what is that, 3.5 months. Where we sit today with the changes in kind of immune checkpoint therapy, this playing field has really evolved incredibly. Thanks to the likes of Jim Allison and other folks here, there are now people that are surviving for many, many years with non-small cell lung cancer. And now with a combination of chemotherapy on top of that, you can actually go from what was essentially back of a death sentence here back in the 2010 to a significant here. Developing a drug in this arena is really going to be tough. So imagine what it's going to be like in non-small cell lung cancer in 2025, if that's when you're trying to get your projects approved. And also think a little bit about what you're actually trying to treat. To move into a life-threatening disease is very different to treating diabetes or treating cardiovascular disease. Projects out there that are really aimed at populations now of millions of people here, the Food and Drug Administration and the European Medicines Agency are really going to set a very, very high barrier here for your projects. They're going to really want you to be paying a lot of attention to a safety profile. They're going to want to actually see good efficacy because they don't want to expose millions of people across the globe to an inferior or substantial project here. And this is a great example here. Pfizer and a number of other groups have moved ahead with JAK inhibitors. They really thought that these small molecule JAK inhibitors could compete against the TNF antibodies out there. And actually, the FDA have made Pfizer and others actually withdraw some of the highest strengths of their compounds just because the safety profile was inferior to those anti-TNFs. On the other hand, things like MS here, we're now seeing projects approved that do actually have safety signals out there. But because you're working in a life-threatening disease here where there's no good cures, the agency are actually a little bit more lenient on those projects to move ahead. So coming back and really focusing on what does it actually make to make a project successful here, essentially you need three good pieces. The first is, as we just mentioned, some disease-relevant biology. To really have some good grounding in your project, that your biology will actually modify unmet medical needs here and have a direct impact on the disease biology. The second piece is really around having a quality therapeutic, something, whether it be a peptide, small molecule antibody, that has all the right attributes to be safe, to be efficacious in humans, and actually modulate that target for sufficiently long to be efficacious. And the third piece is really a focused clinical plan, really focused around unmet medical need. You need to be able to do that experiment in the human and demonstrate it's going to work. So really think about these three key aspects, and if you want to commit to having a drug discovery program, you've got to get all three pieces of these right, otherwise your program is not going to succeed. So thinking about these each in turn, really around the disease-relevant biology, you've got to understand how the disease process evolves during humans. Clearly, in the case here of this particular slide in rheumatoid arthritis, there is a progression over many years, and to come in here and have a therapeutic intervention early on in the disease is going to be very much different from having a disease-modifying agent when you've got late-stage disease. So you obviously need to be thinking about when to treat. The second piece here is really to understand the underlying signaling transduction pathways in a cell, within a multicellular organoid here. How are the various different cells talking to each other? And we also need to understand the relevance of the biology both in a normal tissue and in the disease physiology here. That will help you understand the safety profile of your particular project. And then also thinking a little bit about dependencies. Just because you're treating rheumatoid arthritis doesn't mean to say that's going to be the same in every single individual here. You want to think a little bit about patient stratification, especially in diseases such as oncology. And again, keep bashing on this drum here. Really think at all times in your project about validating your project and your underlying hypotheses here. You really want to make sure that all your thoughts are aligned right the way through the whole of your project. So thinking about the therapeutic modality itself, we really want an agent that is going to have all those effective attributes that Michelle just mentioned to actually make sure it's going to work in the desired manner. So we need to think a little bit about kind of where does your particular target expressed? Where is it actually residing in a cell? Is it residing in the nucleus? Does it actually need to get into the nucleus of a cell? Is it a secreted protein? Do we just need to be modulating that target in the extracellular space? And where particularly does it need to get? Does it need to get up here to the brain because you're treating Alzheimer's disease? Or is it kind of you're trying to modulate the target in the liver or even in the GI tract? It's a lot easier to get your drug there. And then obviously, coming back to Michelle's pyramid there, we need to think about potency. We need to think about selectivity. We need to think about the PK profile. So how long do you need to be modulating the target for? How quickly do you want to get your compound to working? How long does it need to be there? Do we need sustained exposures? We need to be thinking a lot about the safety profile of a particular agent here, and also about how we're going to administer the compound, something that we're going to be giving to millions of people and they're going to take away in a pill bottle here is very much different to something that you're going to be given in an ER here. And also about dosage. Some products are very, very expensive. They have a very high cost of goods, whereas other things that you may be administering, you really want to have a lot cheaper cost of goods. And there's many more things to be thinking about that we just don't have time to touch on today. And then thinking very much about the clinical plan and the medical needs. We really need to be thinking about having that well thought out experiment. We're going to come back to some of these points in a couple of slides' time, but thinking about doing good science, really showing that you've got your compound in circulation, it's modulating its target, and that's actually causing efficacy. Thinking about stratifying patients and the quantitative endpoints. So with that, let's kind of slow down a little bit and step through the three key pieces with a little bit more calm here. And again, these slides are going to be available, so hopefully everybody can refer to them at a later stage. So what really makes a good drug target here? There's a couple of key pieces. One is really around relevance. So really have a good, well-grounded understanding in your particular target. That the biological function, including its substrates, its signaling pathways, are fundamental for disease. Really have some good genetic underpinnings around your data, and ideally, in an ideal world, some good cross-species data to suggest that your particular protein target is going to modulate the biology across species here. And again, banging on that drum that you've got strong validation in multiple model systems that you're kind of modulating that target is going to change that biology. We also need to think a little bit about tractability. Do we think that this is actually a tractable enzyme? Do we think that this is something amenable to a small molecule? Or do we need to be thinking about a genetic reagent because we're targeting a non-coding RNA or something? Coming back to what Michelle was saying, we need to put in place a reliable screening cascade. We don't need to be over-engineer it, but it needs to be fit for purpose here. And we obviously need access to the appropriate preclinical models. If you don't have these pieces, there's no way you're going to be able to execute on a drug discovery campaign. Again, starting early on and then continue to thinking about that clinical development path. Are you going to be able to access those patients? Are you going to be able to be doing the trial in 3, 4, 5 years' time when you're ready to be executing that clinical plan? And then thinking about issues that could arise. Safety is one that kind of trips up a lot of programs, so really thinking about the therapeutic window. But also think about where else everybody is working. There's no point in you working on a target if there's 15, 20 large pharmas out there already working on that target. You might as well go and do something else because they've got a lot more resources than you. And then thinking about key decision-making experiments along the way. Learning from our physician scientists out there, really thinking about kind of conversations about how your particular protein target is implicated in disease, talk and understand where in the disease process it comes into play. There are opportunities for modulating your target in a variety of ways. In this day and age, we can do a lot of genetic validation, either with RNA interference or CRISPR screens. These are very easy to execute, but we also have to remember that a small molecule or protein therapeutic could be different. You can modulate a particular target with a tall molecule, a tall antibody here, but it could actually take you a lot of time to go ahead and develop such an asset. You just need to remember, but both of these are really subject to off-target effects. So really do not wear those rose-tinted glasses. You need to understand what your reagents are and are they actually working on target? Are they modulating the biology in the desired way? And if you go in with flawed reagents, you're going to get flawed results and you're going to delude yourself. And another important thing here is use multiple overlapping reagents here. Don't just base all your validation on one particular oligonucleotide or one particular small molecule. Again, that you could be observing on-target activity, but you could also be observing off-target activity. And then again, get access to patient samples. It's one thing to be working in a cell line that's been cultured in the lab for the last 30 or 40 years. You really hear it when we're in the Texas Medical Center. You want to be going out there and getting access to clinical samples and really thinking about testing your biology in those clinical samples. And really asking the question, is your particular biology pathway of interest deregulated directly in those patient samples? And finally, are you actually seeing what you want to be seeing? Are you modulating that target, and is it having an impact on disease outcome? So just remember, you're not the only person working on a particular target. There is a lot of data out there already. There is a lot of GWAS, gene-associated studies out there in millions of subjects across the globe, and those actually often point towards disease-relevant targets. Frequently, in rare indications, there are kind of a small isolated number of patients that have a mutation in a particular target. And that can really be a laser focus for you to think about kind of how that could make a good target. As we just mentioned, think about cross-species genetics. Is there a connection across multiple organisms here that could kind of point you where to go for human disease? Leverage some of the data that's out there. I mean, there are a bunch of consortiums out there today throwing out kind of whole genome RNA or CRISPR screens. Leverage that information and ask questions about your target. And go out and listen to seminars, listen to other people that are working on the same thing as you, because you could actually kind of, your collective data may point you in a certain direction. I'm not going to speak through this slide at all. I'd really encourage you to go back and look at this slide in the handouts here. But again, remember, don't just work in 2 or 3 cell lines or model systems for your particular target. Yes, that may be sufficient to get your Nature or your Science paper accepted. That probably isn't enough to really kind of convince you to do a clinical trial. There is heterogeneity across the human population. So go in there and test multiple different systems there and modulate your target in both gain and loss of function studies. So what happens if you overexpress your target? What happens if you knock your target out? Does it do the same thing, and does it really point you? Is the biology changing in the way you want it to be? And finally, don't see what you want to see. We all have the habit of scientists of looking at the data and saying, yes, that validates my hypothesis. Do the opposite experiment. Go out there and kind of just ask yourself, am I actually kind of deluding myself here about my particular target? You want to know that before you do spend hundreds of millions of dollars on a clinical experiment. So pivoting to the second point here around having a quality therapeutic modality. So there are many ways of modulating a target out there, and each has its own pros and cons, and this itself should be its own seminar series. You could go in and modulate, and there are approved peptide therapeutics out there. The challenge with peptides is their stability in vitro and also the ability for a peptide to actually get in cell and modulate a particular protein target within a cell membrane. Small molecules are good. I'm a small molecule guy, but there's a significant development time associated with them. And it may take you 1,000 compounds before you get everything tuned into one particular molecule. Antibodies, in this day and age, it's relatively easy to develop an antibody or an antibody fragment. However, antibodies only really work well in the extracellular space. And once you get into development, they can actually be quite expensive. We were having a discussion at dinner last night. It probably costs you about $12 million to be able to jump from an in vitro experiment to be able to test an antibody in the clinic. Again, other protein therapeutics are emerging fields, and cellular therapeutics, again, emerging fields, but there's also very tight regulatory interactions around modifying cells and injecting those back into humans. Genetic reagents are clearly developing, and there are also antibody drug conjugates, cellular conjugates, and this just leads a level of complexity. These may well be fantastic medicines in the future. There's just a kind of quite a high hurdle to kind of get those to the clinic. So this next slide is really going to be a talk that we're going to have, I think, on the 13th of June in this auditorium, really thinking about target product profile. So here, something that you really need to anchor your programs around. Really think about what you're trying to build here. So does it particularly modulate, can you modulate a target in vitro? What do you actually have to engineer into a cell, and how do you think about potency and modulating your targeting a cell? Can you actually develop a particular therapeutic with good selectivity and think about on-target versus off-target activity? Thinking about the PK profile that you want to achieve here when you actually come to treat patients. And then really, do you actually have a relevant activity in vivo here? And finally, about selectivity. So really, as you start developing your therapeutic, really map out this target product profile of all the things that you want to achieve here, and then go through and make sure as you're developing your molecule, does it tick all the right check boxes here? Michelle's already spoken to you around having a disease-relevant screening funnel, echoing a few points that she made here. Really thinking about kind of testing the quality of your molecules, making sure they actually they modulate it through orthogonal experiments here. In particularly, it's very easy to get a down signal. So when you try to configure your assays, think about having an up assay here as well. And another piece that Michelle didn't touch on, think about the quality of your reagents here. Just because somebody sells you a particular protein from a vendor doesn't actually mean to say that what you're measuring is that particular protein here. I know examples of HTS campaigns that have been conducted where it was actually measuring an impurity. So pay attention to the quality of your reagents. Michelle's already spoken to this slide, so I'm not going to talk to it. And I note again here the same figure that Michelle had in her slides, really thinking about these pains compounds. Make sure that your molecules are behaving the way they want to. And so moving ahead in here and thinking about drug testing, there's no right or wrong system to be able to test a particular molecule or protein. All of the model systems here, going from cell lines to PDX models or GEMs, are all perfectly good systems for you to be able to test your disease-modifying hypothesis. Some on the left-hand side of this slide are great because they're simple, they're cheap, you can do them very easily. Others over on this side of the slide here are really a lot more elaborate to do. You may only do once in a lifetime of a particular project, but it's definitely worthwhile going here and testing them. Whenever you do these experiments in whichever of these model systems here, really think about what we call the pharmacological audit trail. Again, this is an important slide you should take from today. Thinking about kind of what concentrations of a drug are we actually using? And is that particularly modulating the protein target of interest? Is that giving rise to functional pharmacology? And then are you actually seeing a relevant phenotype here? So this particular article was authored by a number of authors at Pfizer. There are various different themes about it, but making this audit trail, so compound concentration, biology, phenotype, is fundamentally important. And just to show an example here of a program, the PARP inhibitor program, we understood plasma concentrations of our compound in vivo. We were able to show that we are modulating PARP activity here in both in peripheral blood mononuclear cells and in the tumor. We were seeing DNA damage, and we were able to connect that really with efficacy. So really think about that pharmacological audit trail for your own projects. So all I want to say is things get worse as a program moves ahead here. The first time you make a compound and you test it in vitro, it looks great. However, with time, things will go wrong here. You will pick up warts with your project. That is perfectly fine. There is no such thing as a perfect drug out there. All drugs have flaws. They are compromises. But just ask yourself, how bad is that compromise, and is it stopping us modulating the activity there in patients? And get it right the first time. You're going to spend a lot of time developing a drug. We just want to make sure that you've got the right one out there. It's probably going to take you multiple years to get your hypothesis to the clinic, so we might as well get it right the first time. Optimize your therapeutic. Just because the first molecule you make, the first antibody you make looks good, it doesn't to say that's the one that's got all the right attributes here. I know examples where the 9th molecule made in a particular program was the one that went on into the clinic. I also know programs that took about 10,000 molecules before we actually got the right one to move ahead into the clinic. So be patient and get it right. So jumping ahead, really, thinking about what the next stage is. The next stage is you want to get your program here into the clinic. So have a conversation with the FDA early on. The FDA is not there to be against you. The FDA is there to really work with you. Seek to have a pre-IND meeting and engage in a dialogue with the FDA. They explain your plans to them, what you would like to do, where's your project coming from, and then ask them questions and get them to get some comments. They will really come back. They'll engage you with the dialogue. They'll tell you what they expect to see from you, where their concerns are. They may well get you to pivot your program as well, and your program will be stronger. The first program IX took into the clinic, we had a really good dialogue with the agency, and our clinical trial came out much stronger as it came out. And they will also tell you what to watch out for. If other people have taken agents into the clinic, they won't tell you about them, but they'll also ask questions that will help you get some idea of where they want to go. When you're in the IND-enabling space, this space is really, really highly scripted. There are good guidelines out there, and they're good out there for a reason. So really, don't try and take shortcuts here. The agency won't let you take shortcuts. And they'll put you on clinical hold if you don't comply. Those components of the IND package really around a comprehensive data package. They're really around safety and toxicology, and are really around the drug substance and the chemistry manufacturing controls out here. And you have to get these pieces right, and you have to have it out scripted there to the agency. And then you also need a clinical plan. So jumping ahead to that final checkbox here around the clinical plan, this is where you really get to test your ideas. It's really the time after all of the effort that you've put into here, you actually get to show that whether your program is going to be disease modifying. So it's getting you to the finishing line. But as is going to get spoke later in the talk today, that is where really when we have a value multiplier here. The value of your program goes up incredibly. So don't take shortcuts at this stage. Really make sure you get it right. And the goal here is to eliminate as many variables as possible. Number one, you need to find a like-minded individual, somebody out there in the clinic to work with who's going to be engaged in your project and is going to partner with you to make sure that you can actually succeed in doing the right human experiment. You want to find somebody out there that you can really trust your baby to, and they're going to become your partner in making sure that is successful. You want to run a clinical trial at as few sites as possible. You want to iron out as many possible variabilities out there. And you also want to make sure that those trial sites can actually put enough patients on trial as you want to be able to do. And you really want to think about harmonizing that study population here. If you have a patient stratifying biomarker, you want to use it. You want to make sure the patients that you think are most likely to respond to your drug are actually the ones that you do your clinical study on. Beside that, you really want to think about what is the right endpoint you want to be measuring out there. Ultimately, somebody is going to have to pay for your projects out there, so you want to know what the customer is wanting to see. What are the insurers going to pay for? What does biopharma want to see there? Some diseases, there are surrogate endpoints. They may be good enough, they may not be good enough. We really want to be thinking about using quantitative endpoints. There are a number of indications out there that you may get upwards of a 30% placebo effect there. So if you've got quantitative endpoints, you can really get away from that high placebo score here. And also statistically power is fundamentally important. The last thing that you want to do is do a clinical trial on 50 or 100 patients and not be able to see a signal there. So work with biostats. Biostats are important, and make sure that you power your clinical study accordingly. Skip through this slide in the interest of time. And at the end of the day, somebody needs to be out there to really make money from your project. That is perfectly okay. That's kind of that's the way it needs to go on. Or you need to find somebody out there that's a really wealthy benefactor that's willing to support your program here going forward. The payers and insurers really need to be able to think about how that's going to have an impact on the total cost of healthcare. You could be out there with a CAR T therapeutic that you may be able to charge somewhere of the tune of $400,000 per treatment. At the end of the day, you're able to charge those prices because it's curative. Other things that are supportive care here, what you can actually charge are a lot lower because it's really seen as just an add-on here to the cost of healthcare. And we also need to think a little bit about whether there are going to be barriers to entry for your particular project. At one stage in my career, I worked on a basal cell or smooth end antagonist here for basal cell carcinoma. Seemed like there was some good sound biology there, but at the end of the day here, basal cell carcinomas are typically removed by dermatologists. Dermatologists are paid per surgical intervention. Those dermatologists don't want to prescribe an oral therapeutic because it's basically getting them out of a job here. So there is a significant barrier to entry there. So it's not all doom and gloom. I think the point I really want to leave with you there is that if you do good science, you could have a significant impact on human disease, and obviously profits will follow from that. It is a process. Really, to get one approved FDA therapy there, it is an evolution. It's going to take time and it's going to take resources, but good luck.